prokaryotic cell vital. Just wanted to do a quick overview of them and anatomy of a bacterial cell. So remember, prokaryotic means pro meaning before, karyo referring to nucleus, so this is before nucleus, meaning these cells do not have a nucleus. So bacteria and archaea are the two domains that we will be talking about. So bacteria come in different shapes. Okay. They can be rod shaped, which is also known as bacillus. So rod shaped ones are kind of oblong like that. They can be round or spherical. Those are called cocci. And then they can also be spiral shaped. And those we call spiralia. Okay, and those would be kind of corkscrew shaped like that. Okay, what makes bacteria unique? A lot of things make them unique, but some key points to remember: bacteria have cell walls. Animal cells do not have cell walls. Plants have cell walls, but animals do not. The polysaccharide coat can also be present. That polysaccharide coat can be sticky, so it helps the bacteria stick to its host. Okay. With respect to archaea, now, they can look like bacteria. They can have similar shapes. They even have some weird shapes like triangles and squares and things like that. But so they can be rod or round shapes, but their composition is different. Their cell wall has different biomolecules that make them up. They have different ribosomes. Remember, ribosomes are those little workbenches for protein synthesis. Matter of fact, Archaebacteria's archaea, their ribosomes look a lot like eukaryotic ribosomes. Okay. All right, so now let's go ahead and take a look at what a basic bacteria looks like. So bacteria have a plasma membrane, and remember the plasma membrane, I'll abbreviate PM, that's the phospholipid bilayer. Okay, and it also will have proteins embedded in it. It's the phospholipid bilayer. Right? It's going to have proteins. It may have sugars. And all the things you think about with the plasma membrane. Remember the term fluid mosaic. It's fluid because it can move easily, and mosaic because it is made up of a lot of different components. Okay? So it's got this plasma membrane, and then it has cytoplasm, which is inside of the cell. And the cytoplasm is made up of fluids, so water and ions, biomolecules, okay, and then whatever other structures are in there. And we'll talk about these structures right now. Okay, so let's talk about where their DNA is housed. I'm going to draw this dotted line around here. This is not a structure that I'm drawing. I'm just drawing an area. The area where you find the DNA is called the nucleoid. Okay, and the DNA in a bacteria is a circular strand of DNA. I'm not going to draw it round though. I'm going to draw it kind of squiggly, long, but I'm going to end it together so we know it's truly a circle. It's truly a circle if you draw it right. Okay, but it's all kind of tangled up there in my picture. Okay, that is the DNA. The reason I want to draw it looking different is because bacteria also have another type of DNA called a plasmid, and I'm going to draw this one perfectly round here. That's the plasmid. This is more DNA, but plasmid is DNA. I'm going to redraw that, make this a little bit bigger. A plasmid is DNA that can be transferred. Okay. All right. So that's there. You'll also find in our bacteria, we will find ribosomes. So draw them big dot, little dot next to each other. Big dot, little dot. So ribosomes are made up of two subunits, a small subunit and a large subunit. For some reason I'm having a hard time getting my subunits to connect to each other. So we can draw those around. Again, remember these are workbenches for protein synthesis, so this is where the bacteria is going to synthesize its proteins. Remember a key important feature about bacterial ribosomes is that they are different than eukaryotic ribosomes. Therefore, we can target them with antibiotics. So they're one of those targets we talked about in class. Okay. You'll also find some membrane system in here, and on that membrane system you might find some embedded proteins. This is rudimentary, but this bacteria has a lot of things to do, so it does need metabolic pathways, and so those metabolic pathways have to be organized in some way. You may recall I said this was like having a shelf to put stuff on and keep it organized. So these proteins might be part of a metabolic pathway. Okay. All right, this little guy's got to get around, so he may have some flagella. Flagella help a bacteria swim. It may have to stick to stuff, so it may have these little projections sticking out that are called pili. It may have one in particular that is used for transferring the plasmid. That one's called the sex pili. Okay, I'm just going to draw it bigger so it looks different than the other ones. So that's how this bacteria takes that plasmid and inserts it into another bacteria. Remember, the plasmid was what I talked about with respect to antibiotic resistance. It's the one that can be transferred to share that gene. It's also the one that I talked about when I said that researchers like to use plasmids because they can engineer these plasmids and make this bacteria produce a protein. Okay, that's it. If you have any questions, feel free to give me a call.